Okay, welcome back. I'm glad to be here with you again and tell you a little bit more about the plants and the flora of the Hawaiian Islands. And this is continuing on the same thing as bad taxonomy kills. And I gave a presentation on that a little bit earlier, and hopefully you've seen that already. This situation here is even a little bit more extreme than the last one. This is involving the genus Cryptocarya. It's in the Lauraceae family. You might know the Lauraceae more because it's the family that avocados are in. And the plants that we're looking at today look kind of like little avocados in that respect. And the vegetation looks very much like an avocado tree also. The situation with this species, or this group, I should say, is there was a single species that was originally recognized on the islands of Kauai and Oahu, and that's Cryptocarya manii. It's the only two islands that this genus is recognized on. It's a pretty widespread genus throughout the tropical regions of the world, but we only have this species here. Over on Kauai, the populations were pretty common. You could go into a number of different forested areas, mesic forests, and find small clusters of trees here and there. So it's not considered endangered or anything like that over on that island. On Oahu, by contrast, there is just a single plant in the Waianae Mountains. And it's interesting to go back over at Bishop Museum, where they have a herbarium. A herbarium is a museum where they keep plant collections, and different people that will collect plants will deposit them in the herbarium. And you can go back and see plants that might be collected 200 years ago. And they have those in the herbarium, and they still actually look really good. Some of them still even have the color associated with them. It depends mostly on how they were dried down and prepared. But what's interesting is that you go and look at the Cryptocarya manii specimens from Oahu, and you would see maybe 10 collections, and they all say from this one location in the Waianae Mountains, and again from this particular hill in the Waianae Mountains, and this one little hill in the Waianae Mountains. And he realized that they're all talking about the same tree that was collected maybe a couple decades apart. So this is a pretty old tree that's been there for a long, long time. And it's the only tree that is still present on Oahu. There is some morphological distinctions between the Oahu plant and the Kauai plants. The fruit on the Oahu plant is a little bit larger. The leaves are a little bit different shape and size, and so forth. But the taxonomists that were reviewing this back in the 1980s and 1990s, when they were developing the flora of the Hawaiian Islands, they thought that, well, they don't look morphologically that distinct, and so we're going to lump them together into just a single species. And so both the Oahu and the Kauai plants were referred to as Cryptocarya manii. This is a couple photographs of the tree. If you're familiar with avocados, this is what generally an avocado leaf looks like as well. This is the flower, so they're not very showy flowers, as you can see. Here's a close-up of the flower in the bottom left-hand corner here. And you can see that the petals look like non-colored bracts surrounding the stamen that are on the inside, and then the pistil, which is the female portion, is this region right here in the middle. So the stamen provide the pollen, and you can see the anther sacs here on the present on there, and then the pistil down on the inside. So not a very showy flower, and obviously not a very big flower. And maybe if you're familiar with avocado flowers, because there's a lot of avocado trees all throughout the islands here, you might have noticed this also. And then here is the fruit. This is the plant here from Oahu. You can see over here to the left, Cryptocarya oahuensis. This is the name that was originally associated with just the Oahu plants. And then they lumped that back into the Cryptocarya mannii later. But it does look, I mean, this is a little blacker than an avocado gets. Avocados are typically more green and get very kind of dark grayish when they're ripe. But they have that same kind of look to them in shape, and a little bit of pulp around the outside, just like an avocado does. So our interests were to really look and compare the Oahu plant and the Kauai plants and see if they were in fact that distinctive. And so we used a lot of different genetic tests to document this. We did sequence analysis of these individuals. We looked at one individual from every population over on Kauai, and again, the one plant that was on Oahu. We also compared this to some other species that are present in the South Pacific. So we can kind of get an idea also of where in the world the Hawaiian species came from. We also looked at population level analysis. So not just one individual from each population, but we looked at several individuals from each population so that we can get a sense of the amount of variation within a species, and then how different or not the individual from Oahu was to the individuals from Kauai. So here's the results. This is the sequence analysis. We sequenced two different gene regions. ITS is the intragenic transcribe spacer, which you can now forget until you have to use it later on in your life. This is a nuclear region, and it's present in pretty much every eukaryotic organism in the world. And so it's very nice for looking at species relationships and comparing them across lots of different organisms. This other gene region, TRNK intron, is looking at a chloroplast chromosomal region. And so there's lots of interesting variation, and another interesting story associated with that that I don't have time to get into here. But first, let's look at the ITS. And so here you can see at the bottom of this graph, there's the three outgroup species. The one is from China, and then we have these two species from Australia as forming the outgroup. And so it's kind of an interesting story that the Asia appears to be the closest relative of the Hawaiian species here. But when you look at the Hawaiian species, you can see each of the different populations of Cryptocarya mannii. They all cluster together on this one, what's called a clade or branch of the tree. And then the Oahu individual branches out completely separate from all those. So good, strong evidence that the Oahu plant is genetically distinct at the sequence level from Cryptocarya mannii. When we look at the TRNK intron, we didn't look at as much diversity among the populations like we did up here. We just looked at one individual from Kauai and one individual, and the one individual from Oahu. And again, we were able to see that they were different. They did not come out the same sequence. There was some variation from one species to the other. Again, showing differences between them. When we looked at the population level variation, that's shown here. And this is a principal coordinates or graph of the relationships among the species, or among the individuals within the species. Every color here represents a different population. So you can see the number of individuals for each population that we did look at. And there were 1, 2, 3, 4, 5 different populations that we analyzed here for this. And then here's the one individual from Oahu. And again, you can see how all the Kauai individuals cluster down here at the bottom of the graph. And way up here at the top is the single Kauai individual, very well separated from all the other individuals with it that we analyzed. Again, showing that this is a distinctive species not just a population variant from the others. So again, we went through and we categorized the morphological differences between these, and we're able to show that that is consistent as well, along with this genetic evidence that we have. And again, strong evidence that the Oahu plant, and we can say plants now because they have been able, through tissue culture propagation of that one individual, to create new plants that they could plant out into protected areas and try to save the species from extinction. And this one individual, or these individuals from Oahu, are now referred to as Cryptocarya oahuensis, reviving that name that was used previously for this. And what is more important from a conservation perspective is that this group of individuals are now considered a PEP plant species. PEP refers to the Plant Extinction Prevention Program that exists here in the Hawaiian Islands. And this is a program that gives protection and conservation efforts for any species with fewer than 50 individuals, 50 known individuals in the wild. And with this having just the one tree and then a few saplings that are growing near it, you know, it definitely is considered a PEP species. Where before, Cryptocarya mannii has lots more than 50 individuals. I don't even think it's considered as an endangered species at this particular time. And so if they hadn't made this separation between Cryptocarya oahuensis and Cryptocarya mannii, the Oahu species would not have been legally able to be protected. It would have been allowed to eventually go extinct because that plant would have died eventually. And that would have been a loss of the genetic diversity of this particular group here in Hawaii. So it would have been a very, very sad day for that. But now we can protect it. And there's lots of efforts between not just the PEP program, but also the Division of Forestry and Wildlife, part of the state Division of Land and Natural DLNR, Division of Land and Natural Resources, here in Hawaiian Islands. So again, another example of bad taxonomy, which we had before, can kill. And fortunately, in this case, we were able to rectify that so that the taxonomy is better and stronger, and this is now a protected species. I wanted to kind of follow up a little bit on just some of the efforts in conservation genetics that have taken place over the past 20 years or so. I've been doing this kind of work since I came here in the early 1990s. So close to coming up on 30 years here now, pretty soon. And some of the interesting things that we found, I'm not going to go into great detail, but I did want to show you a few comparisons that I thought were interesting. One of the things that we did was we compared a lot of different endangered species and non-endangered species to show what levels of genetic variation that are present there. And what's interesting is that you can make one generality, and that is that there are no generalities that we can assume from one species to the other. And I'll get into that a little bit more in just a moment. There's lots of factors that go into this. One is the longevity. You know, if you have a long-lived tree, chances are that, you know, if you have a few individuals that are still left out in the wild, they're going to harbor genetic variation among those different individuals just because of their long-lived nature. If they were a short-lived species that would interbreed regularly, a lot of that genetic variation gets lost pretty quickly. The other is breeding systems. There's the different kind of breeding systems that you find among different groups can be very important in terms of their conservation. Some things are obligately outcrossing. They have to cross with another individual within the population. Otherwise, they will not produce fertile seeds. And there's a number of different groups in Hawaii that have that characteristic. There's some groups that breed regularly within themselves. They'll self-fertilize, or they breed easily with very closely related individuals. And so when you analyze these, the obligate outcrossing species tend to rely on, or they tend to host a lot more genetic variation, where something that inbreeds regularly loses that genetic variation. So that lifestyle can be very different. There's also anthropogenic influences, how humans have influenced the populations and what's gone on there. And also the habitat and environment are extremely important. We know that from historical records that certain plants that are only known from a certain region. But when we go back and look at past history of a particular habitat type, we can find evidence that anciently some of these species that are now quite rare and restricted in their distribution used to be quite widespread. And that is something to take into consideration for conservation efforts that might be put into place. So here's a graph showing the different levels of genetic variation among all these different plant groups that I've looked at over the past years. And so we can see a number of different families that are associated and all the different species that are associated here. The percent polymorphism is usually a very good record of how much variation is going to be present within that particular individual or within that population or species. And the more genetic variation, the number will range between 0 and 100 because it's a percentage. The highest is going to be 100%. And so it's interesting to compare these things. You would generally expect that the level of variation within a population is going to be lower than the entire species. And so this is a good example here. The populations of Hesperomani arborescens, for example, range from 26% to 56%. But when you look across the entire species, it's a higher number. And we typically get that kind of relationship for most things that we're looking at here. I wanted to point out a few interesting things with this. In this analysis, everything that you see there in red is an endangered species. And what's really fascinating when you compare this is that the level of variation ranges from 0 to 99%. So just because something is an endangered species doesn't necessarily mean that it's lost its genetic variation. And again, that kind of goes back to saying how you have to look at every situation differently and try to find out what's going on, because it could be because of the breeding system, could be because of how much inbreeding has taken place, its ability to persist with inbreeding, and so forth. So, you know, some things are low, some things are very high, and a lot of them are somewhere in the middle. Even the things that are not endangered, like you can see Acacia koa here, when you look at individual populations, some of those populations have very little variation, some of them a little bit more, and across the entire species it can be quite high. And so you're going to see a lot of differences from one group to another group. A couple interesting stories here. So these are three very different kind of stories of what's going on here. Argyriziphium sandwichensis, that is the Hawaiian silver sword. If you're familiar with the silver sword, it occurs on 2 islands, up at Haleakala on Maui and up on Mauna Kea over on the Big Island. And in particular, this is the Mauna Kea silver sword. So it's a different subspecies from the Maui one. And as you would expect, this is a species that's going to have much lower polymorphism that's associated with it. And in general, they have found that they had to, this is a species that requires outcrossing. It does not self-fertilize very well. And the number of individuals within the population at one time crashed down to literally just a few individuals. And so they've had to go back and hand pollinate a lot of the plants to make them viable so they could produce seeds. Now they have hundreds of individuals that are outplanted because of the conservation effort that's been put into place for these. And the level of variation across the entire species is still quite low because of this bottleneck that it went through. But at least there's lots of individuals out in these outplanted sites so that they can cross with each other when they become into flower at the same time. This species is just the opposite, Cyanea superba. It has absolutely no genetic variation that we could find among the individuals of that particular species. It's an interesting story because it was a plant that was occurring in the Waianae Mountains. It was down to 2 individuals when I first moved here. There was just the 2. And they went into fruit and they collected seeds from them and started growing them in greenhouses. And they found that a couple of years later that those 2 individuals both died. And so this was a species that was growing only in greenhouses and not any plants represented in nature. So this is a really good example of a PEP species in that sense. What they found was when the seeds would germinate extremely well, typically had 70 to 90% seed germination. The plants grew quite happily and they could grow them up quite well. And later on, they started to outplant these into various locations around the Waianae Mountains in fenced exclosures so that they could have nice, good, large populations. And there's several hikes that I go on regularly where I see these when I get up into the mountains there. And so it's always a pleasure to see them. They have a beautiful flowering inflorescence. But what we know from this particular species and this lineage of Campanulaceae is that they are primarily inbreeding plants. And so they have reduced the amount of variation present within the population to 0, as we found here. But they still are quite active and do quite well. And so the opposite story to what we saw with the silver swords that are obligate outcrossers, but they still maintain some level of genetic variation. Coquilla cookii is another really fascinating story too. It was an individual from Cook's Ranch over on Molokai, and there was a number of plants, and then in the early 1900s, and then they started to die. There was just a single plant that was remaining. The person that was running Waimea Botanical Garden on Oahu recognized this as a problem, and so he grafted a branch of Coquilla cookii from the one remaining individual onto the rootstock of a related species, Coquilla drynereoides. And then the host plant died. The original plant on Molokai died. And so the only individual of this species was a grafted branch on the rootstock of a related species. I don't think you get any more rare than that. That's about as rare as I think anything could ever become. What's interesting is that there is still 16% polymorphism. Carrying on that story a little bit more, they were able to get it to grow to maturity. It flowers and it sets fruit. The fruit has seeds that will germinate. The seedlings were growing up to about maybe 2 feet tall, and then they die. And that happened over and over and over again. And when we measured this, there was still, like I say here, 16% genetic variation among these seedlings that were growing up. Again, with the Malvaceae family, this is the hibiscus family. Typically, these are outcrossing species, and they rely on outcrossing to maintain genetic diversity. And again, a story more like the silver sword at the top, very opposite from the Cyanea superba story in the middle there. It required genetic variation, and it just doesn't have much left, and that's why the seedlings don't persist very well in cultivation. Some other interesting stories along this line are these 3 species here. Hesperomania oahuensis. This is a species that occurs in the Waianae Mountains. And there are a few individuals left of it. It's very rare. But interestingly, there's 72% genetic variation or percent polymorphism within this within the species among the different populations, the three populations of the species. Even more extreme are the euphorbia species that we see down here. Euphorbia kuwaliana, which occurs over near Waianae in West Oahu, has about 90% variation, polymorphism within the populations there. And Euphorbia scottsburgii, which only occurs in 3 locations, two locations on Oahu, on Molokai, 1 location on Oahu, and within the populations, 97% genetic polymorphism, and across the species, 99%. Yet it's an endangered species, and going through all the issues associated with endangered species, yet this one still maintains an extremely high level of genetic variation. And when you look across everything there on that list, by far the most genetic variation of anything we've looked at so far. So this really highlights the importance of not just assuming that because a population is rare, because a species is rare, it might only have 1 population and sometimes even just a few individuals in that population. But you know, it could have lots of genetic variation, it could have none. And it's important to understand that. If we understand what the level of variation is, what the breeding system is with this, what some of the requirements for growth of these species in larger populations might be, it really gives managers a much better idea of how they can help these things to survive and persist during difficult times for their growth. So I want to thank you for your attention and wish you all the best. Thank you very much.